Okay, in this video we're going to be talking about a couple of different things. Um, just a review from last lecture. Transport RNAs, they make up 15% of the total cellular RNA, and they bring the amino acids to the translating mRNA. They're small, but they're small sequences, but abundant. They're less than 100 nucleotides long. And the ribosomal RNA, they are 80%. They make up the bulk, which is emphasizing the major part of protein synthesis in the cell. So 80% of the RNA in cells are ribosomal RNA. And they are between 120 and 5,000 nucleotides long. They range from like 5s, the Swedwick number, to 28 Swedwick number. And the messenger RNAs make up 5%, and they are very different in size. They can get from 500 to 10,000 nucleotides long. And the genetic code has 64. You get that by multiplying 4 times 4, 16, 16 times 4 is 64. So because you have 4 different nucleotides and you have 3 letters in the codon, so that's why you do 4 times 4 times 4. And 61 code for amino acids, so there's some redundancy. Some amino acids can have up to 6 different codes, and there are 3 specific stop codons, which are UAA, UAG, and UGA, and 1 specific start codon, which is AUG. We always write 5' to 3'. The wobble has to do with the third position. A lot of the amino acids does not change if the third code is changed. That's basically the wobble. Codons that differ in their first 2 base pairs must have their own tRNA. So here's a little dictionary. So we see that serine has 6 different codes, and there's no scientific explanation why some have only 1 code, some have 6. It's just the way it is. And here's a way to like look up something. Let's say you want to look up AUC. So you see A, and all these begin with A, and then U. Actually, all these begin with A, all these amino acids, and then all these amino acids begin with AU, and then let's say you're looking up AUC, then AUC. So it's isoleucine. Then you could look up anything else. So GGA, so G, and then GGA, so GGA, glycine. So, and here's a little example of insertion deletion. So let's say you insert a C here. That's the normal RNA, and every single codon is changed. So over here you have this, and this is conserved, but then you have added this. So the UAC became UCA, and then this gets changed and keeps on changing all the way down because you added 1 extra nucleotide. Same thing with the deletion. You delete 1, and the whole code changes here on down. Here on down. From here on, everything's changed. GCC, CCU, UAC, ACG, GGA, GAU. So all these amino acids are going to be different and they're going to keep on changing. And then, but if you have a addition and a deletion, it's going to sort of balance out. So you get like two wrong amino acids in this case, and then it's correct over here and correct over here, and it's going to be correct down the road. So this one's a little bit less serious, but still you have two amino acids. So it depends on the protein's structure. If it's the structure gets changed a lot, the function will be um, function might be messed up. Um, and then over here, they're showing the effect of terminating codon. So you have UAA, UAG, and UGA. So over here we're saying UAA. So UAA here, and then another UAA here. So it's ending early. So over here also, it's ending early instead of going all the way down. Um, the anticodon and the codon over here. We have the anticodon because we're reading it 3' to 5' UAG, and then on the codon you have AUC, and then there's some there's only 30 to 40 transfer RNAs, so some transfer RNAs are going to be able to recognize more than one code because there's 61 codes and 30 to 40 transfer RNAs in a cell. So there's like 30 or 21, 30 to 21 transfer RNAs missing. So that problem is solved because the season A's they have a specific one, but what U and G they have two different ones. So the U can bind to the U in the anticodon can bind to a or a G in the codon, and the G in the anticodon can bind to a C or a U in the codon. And there's inosine also, which is a deaminated deaminate nucleotide that can bind to the A, U, or C. So the third base can be changed, can be the third nucleotide in the codon and the anticodon. There's some variability. And then we have missense mutations, nonsense mutations, insertion and deletions, splice mutations. The splice ones would be we have a problem with the AG and GU and then you might have an extra bit of intron in the exon, or you might chop off a bit of exon. That would be splice mutations. And frame shift, we talked about that before, how you delete one letter, one code, and then all the triplet codons are messed up. And nonsense mutations is when you have a stop codon put in UAA, UAG, or UGA, and then it's going to stop it early. If it's like almost all of them in the end and you're only missing one or two amino acids, maybe it's not a big deal. But if it's missing a whole bunch of amino acids, the structure is going to be very different, and the protein might not work good. Missense mutations. That's when you have um, different amino acids. Sometimes it could be very bad because the amino acid might be very different and the structure might change completely. Or in other cases, it might not have a big impact and it might be alright. Silent mutation is when you change the last letter usually, and the amino acid remains the same. The protein's function would be the same, but the the code might be mRNA might not be cut up by the microRNA because microRNA hydrogen bonds with this with mRNA, and to see if it's the right mRNA it needs to cut up. So there might be an extra aggregation of the mRNA because it's not being able to be cut up by the microRNA. Um, amino acid and transfer RNA synthetase. This is where amino acids are going to be bound to the transfer RNAs. Um, amino acid is activated by the reaction with ATP to give an amino acyl AMP intermediate. So that's what's going on over here. So ATP is being used and two phosphates are leaving, and then over here this OH is going to leave, and then we're adding a P ribose adenine, and then we're gonna add this to the transfer RNA up here. This is where the CAA is. So on the adenine, the OH group on the CAA, on the A, it's gonna get added, and I need to ester linkage, A residue. The enzymes must be very precise, so are bridging between three-letter codons. So the enzyme tRNA synthetases, those are the ones that are gonna make sure the right amino acid is being paired up with the right tRNA. There's 20 enzymes for the 20 different amino acids. Um, the so this is the tRNA synthetase, and if there's an incorrect amino acid, then the amino acid will be removed from the tRNA. Here's another diagram showing tryptophan being added to tRNA. This tRNA is supposed to be carrying tryptophan, and ATP is being used, and then over here on the A residue we have the tryptophan being added, and then over here we have the anticodon and codon locking, and the tryptophan is going to be encoded. The tryptophan is going to be used in the translation process. Um, charged tRNA bound amino acid. Yeah, so over here, tRNA is bound to amino acid, and over here, it's talking about the methionine, the first methionine. So we have to differentiate between the first methionine and the subsequent methionines used in elongation. The first methionine is formal methionine, and so it has to be formulated, and the transfer RNA that's going to be carrying the first methionine has to be different. So the tRNA is going to initiator tRNA, the first charge with methionine, and charged with methionine, and then a formal group is transferred to methylene transfer RNA from N10 formal tetrahydrofolate. So the formal methionine is not going to get added to the while elongating. So the other tRNA for the methionine is different, which does not allow for the formulation of the methionine. And here's just the autologs between prokaryotes and eukaryotes. When the ribosomes, they're not actively translating, they remain as two separate subunits, large subunit and small subunits. And then we have that 5, 23, 16 ribosomal RNAs 25S, 23S, 16S, and their corresponding nucleotide lengths. And over here also we have the different RNAs over here. So this 5 and this 5 is autologs. This 23 and this 28 are autologs. This 16 and this 18 are autologs. So they all have all these different this different sizes of nucleotides. The ribosomal RNA are autologs, which means they have common ancestry. All right, that's about it.